Actually, I also think it's not only the customers, but also the health authorities that have this kind of need or the wish to reduce animal testing in their portfolio for endotoxin testing. So this is why they also change or allow now much more companion tests or alternative tests if they prove to be equivalent to the common LRL assay. Yeah, so for me, it's really, I think people that are now changing from, for example, LRL-based assays to recombinant factor C or monocyte activation tests really have this wish of being more sustainable, being more in peace with the ethical concern about animal testing, like the horseshoe crab bleeding or rabbit testing, so that they can contribute to a change of ethical endotoxin testing.
Welcome back to the Smart Biotech Scientist. I'm your host, David Brulman, and if you're joining us today, we are in the middle of a fascinating conversation with Anika Hoffmann about endotoxin testing. Anika is a postdoctoral researcher in analytical chemistry at the University of Applied Sciences in Sion, Switzerland. In part 1, we uncovered why these bacterial fragments keep quality assurance teams up at night and the surprising limitations of the decades-old LAL test that most of us still rely on. But now comes the exciting part, the solutions. In this second half, Anika Hoffmann will reveal the breakthrough analytical approach that's positioned to disrupt endotoxin testing as we know it. If you've ever wished for more accurate results, real-time monitoring capabilities, or simply a testing method that doesn't depend on horseshoe crab blood, you're about to get a glimpse of the future. So whether you're heading home after a long day troubleshooting in the lab, or squeezing in some professional development during your morning commute, these next 10 minutes could completely transform your approach to endotoxin management. Let's make bioprocessing smart together. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brulman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics at market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. And now as we compare these two methods side by side, so the traditional LAL and your novel chemical method, can you highlight just again what are the main advantages of each and maybe what are still the limitations?
As I already said, for the LRL assay, it is basically its sensitivity. So it's, as I said, it goes down to 0.01 EU per milliliter. It is a fast assay as well. So the measurement itself doesn't take long. It has the side effects or disadvantages of having this low endotoxin discovery effect, being sensible to the matrix the sample is in or the sample manipulation. And this is where our test, for example, we are unfortunately not as sensitive yet as the test, but we are much more robust. So we have our coefficient of variation is for normal samples around 5%, maximum of 10%, depending on how complicated the matrix is. It is also comparable with the time it takes to prepare the sample is comparable and overall to the LAL assay. So, and of course we're not limited to the physiological conditions.
If a company wants to move away now from LAL towards your new method, obviously there will be some regulatory challenges, probably some validation, some comparability studies. What would a company need to do to convince the health authorities?
Yeah, basically what you just said, you need the validation studies to prove that the assay performs as well as the approved assays. So it could be either the LRL assay, which is an approved endotoxin measurement, Or since 2021, for example, the European Union, you have also the monocyte activation test, which replaces the rapid pyrogen test. So to have more variety and to reduce animal testing for endotoxin measurements. So it would also be sufficient to show that it's equal to the monocyte activation test for some applications. Or in Europe, we also have now the recombinant factor C test. So this means that there's no lysate from the horseshoe crab is necessarily used because the recombinant factor C test uses recombinant produced factor C, which is in Europe also accepted. In the United States, not yet, but has to be validated. So if the validation shows it is equal and performs well, it can be also used for endotoxin testing.
What will be the implications with respect to the devices needed for the new method? Because what I understand from the LAL test, it's quite a simple test to run. How would that change with the new chemical test?
Yeah, with the new chemical test, you would need different equipment. With the LAL assay, you have very dedicated equipment, which is, as you said, in its manipulation itself, very simple. So, for example, you have like the cartridge test where you have just to pipette 4 times 25 microliter in a channel, and then you put it into a slit and the automation is run, or the sample analysis is run automatically by the software. For our test, it is a liquid chromatography test. So you would need an HPLC system with fluorescence detection. But once you have this HPLC system, you're not necessarily limited to endotoxin analysis. You can do a lot of different analysis. You can characterize your proteins, you can measure your impurities with this instrument. So it's more versatile, but it's a bit more complicated as the LAL assay cartridge set, for example.
And to what extent would the HPLC allow you to do real-time monitoring of LAL? Would that be possible at all?
So with the current test, no, there's no real-time sample monitoring. What could be a vision, because we have the sample preparation, and until the sample is really analyzed, there will be at least a difference of 3 hours. But what we could imagine, for example, is, because I did some experiments where I followed along a bioreactor for PHA production, where I directly measured the supernatant of this fermentation during the fermentation, and I correlated the results to optical density, for example, and dry cell weight of the bioreactor. So you could set up models that can correlate these parameters to each other, as you can measure, for example, optical density and dry cell weight in real time. Maybe the model becomes so performant that you can deduce how much endotoxin you would have in the supernatant of your bioreactor, for example.
Well, speaking of correlation, how well does the KDO method correlate with the LAL method? I imagine you have done some side-by-side testing.
Yes. So here you have to take 2 things into account. So one is that KDO method is a concentration-based assay. So we have absolute values. We have nanogram per milliliters, for example. And the LAL assay is a biological activity test. So you have the endotoxin units per milliliters. But if you do side-by-side testing, we see a linear correlation between these 2 tests. So it's good, yeah.
Yeah, that's a good thing, definitely. Well, beyond the analytical advantages with your new method, there's also a sustainability aspect. So it definitely eliminates the dependency on the horseshoe crab blood. As you're talking now to customers and people in the industry, what is your impression of how important this sustainability aspect is actually in the biopharmaceutical industry?
Actually, I also think it's not only the customers, but also the health authorities that have this kind of need or the wish to reduce animal testing in their portfolio for endotoxin testing. So this is why they also changed or allow now much more companion tests or alternative tests if they prove to be equivalent to the common LAL assay. Yeah, so for me, it's really, I think people that are now changing from, for example, LAL-based assays to recombinant factor C or monocyte activation tests really have this wish of being more sustainable, being more in peace with the ethical concern about animal testing, like the horseshoe crab bleeding or rabbit testing, so that they can contribute to a change of ethical endotoxin testing. And on the one side, it's a good thing because it's a sustainability, but I think it's also a necessity. Because when we look at development of the horseshoe crab population, we see it's declining. And on the same coastal area destruction and overfishing. And on the same time, we have increase of demand for endotoxin testing. So in a certain point in the future, we would have conflict between the available methods and the life of the population of the horseshoe crab species.
Yeah, definitely there is an issue with this test, and hopefully a lot of companies will also be ready to move over to more sustainable assays in the future. This leads us to the final part of our conversation because you're in a transition now from academia towards entrepreneurship. You have a successful technology. Tell us a bit what you envision, what stage you are with your new company, and when can people start buying, for instance, this new assay?
So I'm in what is called an early stage startup phase. So we're still figuring things out and we're setting things up. So we currently are developing these kind of kits to bring it onto the markets. But especially now we are at the phase where it's very important to have the feedback of customers and also samples that show that our assay is performing as we hope it performs or how it helps people. I think for me, in the startup journey, we are now in a very interesting phase to go to the market phase, I can say.
And what was the trigger for you to take this leap? Because, I mean, it's quite a different world, academia and being in the marketplace. Was this something you have planned, or it was now just an opportunity, or how did that go for you?
No, actually, it wasn't planned at all. So already doing my PhD wasn't planned. It's kind of, I'm following a pathway of unplanned events. It's actually, when I had this culture shock with this endotoxin tests, and as an analytical chemist, I know what analytical methods can do, what we're capable to do. And then we're still using validated tests with uncertainty ranges that are unimaginable for new developed analytical tests. It was kind of, if I have a solution, I will make it accessible to everyone. I want to improve that. And then it kind of all into together. And I was really starting to think how I could make this accessible. And then it clicked kind of, I think I will go the entrepreneurial path for it.
Yeah, that's exciting. Well, I commend you for that. That's courageous. It's great.
Thanks.
And often career paths are not very linear, so there's a lot of unplanned stuff happening. So
Yes.
as you're looking now onto this new season, what is the thing that excites you the most about being an entrepreneur?
Actually, the challenges that lie ahead, setting up the company, acquiring the customers, getting to know new people, getting your network. Yeah, I think it's very different from academia, I imagine. And I'm aware of that. I have still a lot to learn. Basically, we'll have to go through a lot of pitfalls, probably, but I'm determined to overcome them. And I'm really looking forward to it. Yes.
Oh, that's excellent. What advice would you give somebody who is at this similar stage right now, or maybe still a bit on the fence? Like, should I take the leap or not?
If somebody's really at a stage where he or she has an idea and she has identified a market need, then I would clearly say go for it, because I think there's nothing worse than having constantly this question, what if I had done it? Would I have succeeded? And even if it turns out a few months, a few years later, well, then at least you tried. So go for it. Yeah.
I like that. Yes, go for it. Smart biotech scientist. If you want to start a company, if you notice the problem that you want to solve, go for it. Take the risk.
Yes.
This has been really fantastic, Anika. With all what we've discussed today, what is the most important takeaway?
I think the most important takeaway of today's discussion is endotoxin testing is a very crucial quality control for biotechnological products, biopharmaceuticals, and medical equipment. That the current tests have serious struggles concerning matrix issues and endotoxin masking effect, and the future might lay actually in a robust and reliable chemical assays for endotoxin testing.
Anika, where can people get a hold of you and maybe potentially even early access to a kit? Or, as you said, you need customer feedback. So I imagine you would love to have some conversations with potential customers.
Yes, totally. You can reach me via my email or my LinkedIn, which I think will be provided in the description of this podcast. Don't hesitate if you have a question or you're interested. Essay, reach out to me. I'm happy to have this conversation to discuss with you. And also, if I may, I'm currently looking also for co-founders. So if you're somewhere out there and you share the passion for making endotoxin testing ethically correct and without the need for animal product, and really to enable it to bring it to everyone who needs this kind of test, also please feel free to reach out for me.
Well, Smart Biotech Scientists, what an amazing opportunity. I think that's the first time we pitch a co-founder or physician. So please do reach out to Anika. It's definitely a big problem we need to solve, and it will be a game changer in the industry. So thank you very much, Anika, for being on the show today. And I will leave all the links in the show notes so you can reach out to Anika. So thank you very much.
Yeah, thank you, David, for giving me this opportunity to share my research and the journey of endotoxin analytics.
And there you have it. The future of endotoxin testing unfolded right here on the Smart Biotech Scientist. I'm excited to see how this sustainable alternative assay transforms biopharmaceutical manufacturing in the coming years. If you're feeling inspired to implement new technologies, remember that doing so doesn't have to be overwhelming. Book a free call with me at BrullmanConsulting/call to discover how you can fast-track your biotech development without the costly delays. Until our next episode, keep questioning the status quo, stay curious about new technologies, and remember, science may give you headaches, but biologics drug development should not. This is David Brullman signing off, and I'll catch you next week for another dose of bioprocessing wisdom. Take care and make it smart. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.